So here we are in the final lecture chapter of this semester. Congratulations for making it this far. This one's a pretty short one, but we'll be discussing diagnosis of different infections using all sorts of different techniques that we may have been introduced to slightly throughout the semester. This semester, we've been covering a lot of the most clinically significant microbial infections in our course wiki. Here, we're going to think about how we diagnose each of these infections. There are three general ways that we'll look. At phenotypic analysis, we'll see morphology, biochemistry, and chemical analysis. There's also genotypic analysis available these days, as well as immunological analysis. We'll look at a variety of techniques under each of these categories. The form on the right is a form that you might see if you work in a clinic, and you'll probably become very familiar with it. Under the category of phenotypic methods, we can look at the microscopic morphology, as in the cell shape and size, the results of a Gram stain, or acid-fast reactions, which you're familiar with, as well as some special structures like endospores, granules, and capsules. In macroscopic morphology, we need no microscope. We're looking at traits that can be assessed with the naked eye, like the appearance of the colonies as they grow on a gel or in a tube, their texture, their size, their shape, the pigment, and their patterns of growth. We can also look at physiological or biochemical characteristics. The physiological methods are the traditional mainstay of bacterial identification. We use diagnostic tests for determining the presence of specific enzymes and assessing the nutritional and metabolic activities. For example, their fermentation of sugars, how they metabolize different polymers, and what sorts of gases they produce. We can also test their sensitivity to antimicrobial drugs using some of the tests we examined early in the semester. Finally, we could use chemical analysis. In chemical analysis, we're taking the types of specific structural substances that the microorganisms contain, and we're looking at their chemical composition, for example, the peptides in the wall or in the lipids that are in the membranes. In genotypic methods, we're examining the genetic material itself. These techniques have revolutionized how we identify and classify bacteria. The primary advantage over phenotypic methods are that with genotypic methods, we don't necessarily even need to culture the microorganisms. We need just a very small sample. Also, they're becoming increasingly automated, which means that we can get results super fast. Through the use of genotypic methods, scientists are beginning to realize that there are so many more microbes than we ever knew existed, because many of them we cannot grow in culture. Recently, we've discovered 30 new species of plaques alone. You know, the ones on your teeth. Finally, we'll look at some immunologic methods. As we know, microbes have antigens on their surfaces that are recognized by the immune system. So we can use the antibody response to antigens in order to determine what antigens are present. We can use blood testing, which is often easier than testing for the microbe itself. We're just simply looking for the antibodies that our immune system has put there. We're testing for the presence of specific antibodies to a suspected antigen. HIV testing uses an antibody for the virus in order to discover whether a person has been exposed to the virus, because the viruses themselves hide out inside the immune cells for most of their life. And there are a number of different immunologic lab kits that are available for immediate identification of a number of pathogens, which allows us to determine very quickly what might be going on for an individual. So the success of identification and treatment depends on proper collection, handling, and storage techniques, and that is going to be up to people like you. General aseptic procedures in collection, handling, and storage are absolutely imperative. Otherwise, you could end up with contaminated samples. The figure on the right shows you a number of different means that we use to collect specimens. Some of them are from body fluids, or some of them are from direct swabs. In the lab, we can use direct tests using microscopic, immunologic, or genetic methods. Then we could use cultivation, isolation, and identification of the pathogens using a wide variety of general and specific tests. And the results of specimen analysis are entered in a summary patient chart, just like the one you see on the right here. And you'll ask for the specific tests that are necessary from the microbiology lab that you send your samples to, or maybe you do them in-house. So this chart summarizes specimen isolation and identification. Here we have the specimen. We can look at it from two perspectives, the specimen or the patient. If we have the specimen, we can either do direct testing or culture isolation. In direct testing, we might use the gram stain, acid-fast stain, fluorescent antibody stains, or gene probes, and look at them under the microscope. We could also look at the direct antigen or gene probes macroscopically. In cultivation, we're going to be utilizing a lot more of the biochemical tests, serotyping, antimicrobic sensitivity, gene probes, phage typing, and animal inoculation. When we look at the patient's perspective, we can do immunologic and serological tests. We'll examine some of these shortly. We can do in vivo tests for reaction to the microbe, or we can look at clinical signs and symptoms in order to make an assessment of what the pathogen might be. Under phenotypic methods, we can make immediate direct examination of a specimen using gram stains or acid-fast stains or direct fluorescence antibody tests, maybe even direct antigen testing. The staining techniques that are most often employed are the gram stain and the acid-fast stain. They're useful, but they don't work on all organisms. DFA tests, or direct fluorescence antibody tests, highlight the presence of microbes by labeling antibodies on their surface. These are useful for bacteria like the syphilis spirochete that cannot be readily cultured in the lab. Direct antigen testing is like DFA, but the reactions can be seen with the naked eye, just like an ABO blood grouping test. If there are only very few microbes, then direct testing is not really that effective. We need much more sensitive methods, which we'll explore here. If we're interested in cultivating a specimen, selective media are often necessary, especially with samples like feces and urine, because they have really high counts of bacteria and a huge diversity of species. So by using selective media that only perpetuate the growth of the things that we're interested in testing for, we can get a more clear understanding of what might be there. We can use various means of biochemical testing, like carbohydrate fermentation. They produce acids or various gases from this fermentation process. Hydrolysis of the gelatin, starch, and other polymers within the selective media can be examined. And enzyme actions like catalase, oxidase, and coagulase can be examined. We also might be able to measure the byproducts of metabolism. Here's an example of whether we determine if an enzyme is present in the microbe or not present in the microbe. We can have an unknown microbe plus a substrate. If the enzyme's present that turns this substrate into a particular product, then we'll see that there's a product formed, which is a positive result. This may be indicated by a color, or it may just be indicated by the presence of a colony. If the enzyme is absent in that particular microbe, then there'll be no product formed, and thus we have a closer idea of what type of microbe it might be. Now, many of these biochemical tests can be performed with a rapid miniaturized system. In this system, we can simultaneously determine up to 23 different characteristics, just based on the reactions and colors seen in each of these different samples. Different substrates are placed in each slot in order to see whether there's a metabolic product produced, and those metabolic products are measured through color. There are many common schemes that are used to identify bacteria, and they're based on easily recognizable characteristics like how they move, what stain reactions they have, their shape, and biochemical reactions. We form what's called a dichotomous key. Let's say, for example, we're looking at rod-shaped bacteria. Here we are in this dichotomous key, and they're rod-shaped, but do they stain positive or do they stain negative? If they stain positive, we're down in this side of the tree. Once we see they're gram-positive, Are they spore formers or non-spore formers? If they are gram-positive spore formers that are rods, then they're going to be Bacillus clostridium. So these dichotomous keys can really help us identify a different species based on a series of different tests. Often we need to determine the clinical significance of cultures. Is something that we've isolated clinically important, or is it just normal biota? How do you decide whether it's contaminant or normal? Some of the possible criteria are the number of them. And if you can repeatedly isolate a relatively pure culture of any microorganisms in numbers that would suggest that it has become pathogenic. Let's have a quick concept check. Which of the following is not a biochemical test used for identification of pathogens? Is it carbohydrate fermentation, polymer degradation, enzyme activity, or gram reaction? Keep in mind, the key here is biochemical test. And you recall that the gram reaction provides morphological information. Now we'll take a quick look at genotypic methods. The order and arrangement of bases in DNA is unique in each and every organism. That makes it possible to identify bacterial species using DNA techniques. DNA analysis often uses genetic probes or hybridization. That's where we have a particular sequence of DNA that we know, and we can use that to identify a bacterial species by analyzing its segments of DNA to see if they match the known piece that we have. If it hybridizes, as in sticks to the bacterial DNA, then we know that we've identified that particular species. So small fragments of single-stranded DNA are the probes, and those are the things that are used to hybridize or connect to the bacterial DNA. The segments we have as probes are known to be complementary to specific sequences of DNA in a particular microbe. So DNA is taken from cells and bound onto blotter paper. And then we add probes to the blotter and observe for signs that the probes have become fixed to the test DNA. Often these probes have a fluorescence or other sort of dye to allow for their identification. Another DNA technology is using nucleic acid sequencing and ribosomal RNA analysis. You'll recall that we've used RNA in ribosomes in order to determine the relatedness of individuals. So most of the RNA in the small subunit of the ribosome is conserved evolutionarily, and this is the portion that allows us to assess the relatedness based on RNA sequences. However, some regions of the ribosome are highly variable between species, which makes it especially well suited for differentiating one species for another. Basically, we're comparing the sequence of nitrogenous bases in the ribosomal RNA. This is effective for differentiating general group differences and can be fine-tuned to identify at the species level. One of the techniques is called FISH, or fluorescent in situ hybridization. This hybridization works with the 16S RNA. You don't have to recall what that's all about, but we can identify them in 90 minutes instead of the 24 hours that we used to have to wait. In this figure, you can see a simplified version of how this works. We have a positive blood culture tube, and we've gram stained it, so we know that the gram stain is positive. So we've assumed that it's Staphylococcus, but we don't know if it's Staphylococcus aureus. So we can use a probe for S. aureus, and it has a fluorescence molecule on it. If it hybridizes with the DNA from the ribosomes here, then the result is a slide that glows like this. We can see this in situ hybridization of the probe RNA with the RNA of the ribosomes in Staphylococcus aureus. So yes, indeed, in this case, it was. Another technique that's often used in many genotypic methods is polymerase chain reaction. You may have heard of this or you may not. It's covered in detail in one of the chapters that we didn't cover this semester. But it allows for rapid identification of pathogens because we don't need to culture the pathogens. Polymerase chain reaction is a way for us to magnify the amount of DNA that's present. So we can take just a very small sample of DNA and then magnify it by thousands and thousands of times using polymerase chain reaction. Essentially, this technique allows DNA to polymerize or grow and grow and grow, and so we can make thousands of copies in a very short amount of time. And it's used to increase the sensitivity of many of these other tests. It's been developed now for a wide variety of bacteria, viruses, protozoans, and fungi. So polymerase chain reaction would be used along with some of these other techniques in order to generate more DNA and establish what sort of bacteria there is. Let's have a quick concept check before we move on. What type of identification is FISH analysis? Is it morphological, genetic, biochemical, or immunological? Well, so far we haven't covered immunological. Because it involves DNA, it's not morphological, we cannot see it. It's not biochemical because we're not looking at products of certain reactants that are put in there. It involves DNA, so we'll go with the genetic option here.